In our previous video, we delved into the fascinating process of muscle contraction and its crucial role in enabling our movements. The whole process is known as the sliding filament theory, which is mostly used to explain the process of muscle contraction. Let's take a moment to review the entire process in more detail. But before starting the sliding filament theory, let us go back to the smaller details first. It all begins with a stimulus that is provided to the muscle, signaling it to initiate movement. This signaling is done due to neuromuscular junction, and this stimulus triggers the release of a neurotransmitter known as acetylcholine from the neuromuscular junction. The neuromuscular junction serves as the connection point between nerves and muscles, responsible for transmitting signals from the nervous system to the muscle fibers. Here are the muscle cells, and here is the nerve fiber. Now, at the end of the nerve fibers, there are receptors for acetylcholine release. At the neuromuscular junction, acetylcholine is released, setting off a cascade of events. This release of acetylcholine generates an electrical impulse known as an action potential. Now, do you recall what happens when an action potential reaches the sarcolemma, the cell membrane of muscle fiber? The action potential means that the charges of the sarcolemma will be exchanged. The action potential causes voltage-gated channels on the sarcolemma to open, facilitating the exchange of ions inside and outside the muscle cell. At the state of rest, the charges on sarcolemma are positive on the outside and negative on the inside. But when the sarcolemma is in an excited state, when the action potential is generated following events take place. Specifically, the action potential triggers the opening of sodium and calcium ion channels. These ions rush into the sarcolemma, creating a positively charged interior and a negatively charged exterior. This change in charges of sarcolemma sets the stage for two important processes. Firstly, within the sarcomere, which is the basic structural unit of a muscle, the myosin head binds itself to the actin filament. This binding occurs due to the charge difference resulting from the influx of ions, which was achieved by the action potential. Simultaneously, a biochemical reaction takes place. Adenosine triphosphate, ATP, which serves as a primary energy source within cells, is converted into adenosine diphosphate, ADP, and an inorganic phosphate group. These two phenomena set the wheels of muscle contraction in motion. The binding of myosin to actin, powered by the energy released from ATP conversion, prompts the myosin head to move forward, causing the actin and myosin filaments to slide past each other. So, the ATP is converted into ADP and released energy. This energy causes the myosin head to attach to actin. This head moves forward and sliding occurs, and the myosin head detaches. Then again ADP is converted back to ATP, and then again the myosin head attaches to the actin filament. The energy derived from the conversion of ATP into ADP enables the myosin head to attach itself, propel forward, and subsequently detach. The cycle continues as a fresh ATP molecule binds to the myosin head, restarting the process of attachment, forward movement, and detachment. This repetitive action propels the sliding of filaments and results in muscle contraction. Now, you might be wondering about the source of ATP that fuels this remarkable process. how this ATP is broken down and gained back to its original form. If we look at the whole process of muscle contraction, there are three major places where we will require energy. The major energy required will be here in the process of sliding filament theory. This is because the myosin filament has to move along and cause the contraction. There are two other minor points where energy is required: when the sodium channels are required to get opened and when the calcium ions are needed to be opened. These three areas of the muscle contraction process are requiring energy. So, how these three movements are facilitated? Well, there are three major sources of energy involved in these movements. The first source is the phosphorylation of ATP by phosphocreatine. But how phosphocreatine is made? Phosphocreatine, also known as creatine phosphate, plays a crucial role in maintaining the energy balance within muscle cells. During periods of rest or low-intensity activities, when energy demands are relatively low, there is an excess of ATP available in the muscle cells. This surplus ATP provides the opportunity for a process called phosphorylation to occur. Now, what is phosphorylation? It is basically the process by which a phosphate group of ATP is transferred to a molecule. Now, in this case, we are talking about creatine. So, phosphorylation involves the transfer of a phosphate group from ATP to creatine. This process is catalyzed by the enzyme creatine kinase. The result of this enzymatic reaction is the formation of phosphocreatine. So, this phosphocreatine will be present in the muscles with an amount 5 times greater than ATP. Now remember that in sliding filament theory, we discussed that ATP was converted into ADP. Now we need this ADP to convert back to ATP molecule in order to perform the regular sliding filament mechanism. This is where the phosphocreatine will jump in. This ADP will take a phosphate from phosphocreatine and form ATP. This ATP will be utilized again in the sliding filament mechanism. So, what happens is that in a resting state, ATP is converted into ADP, and the phosphate released is taken up by the creatine and converts itself into creatine phosphate. When the muscles are in a contracting phase, this phosphocreatine is then broken down into creatine, and the phosphate group is now taken up by the ADP molecule to gain a new molecule of ATP. The second source is known as the process of anaerobic glycolysis. As the name suggests, that glycolysis is occurring without the help of oxygen. These glycolytic reactions are useful when the phosphocreatine gets lowered in the muscles. Basically, this process is followed by the conversion of glucose into 2 pyruvate molecules with the release of 2 ATP molecules during the process. The details of this process of glycolysis can be checked out on our website as well. Now, these pyruvate molecules are converted into lactate. This process mostly occurs during the absence of oxygen. This process is activated when the muscles are doing intense exercises for a long time. Now, the 2 ATP that were made in the process of glycolysis is utilized to replenish the stores of phosphocreatine. The process of glycolysis obtains many end products. Due to this, muscle contraction is slightly affected, so we need a third source of energy for muscle contraction as well. The third source of contraction is known as oxidative metabolism. Oxidative metabolism is the set of reactions taking place inside the body. The end products obtained with the process of glycolysis are then combined with oxygen and produce ATP. This ATP is then utilized for the process of muscle contraction. So, the main source will be ATP, which will be driving the process of muscle contraction. And obtaining this ATP will be done in 3 ways, which are phosphocreatine, glycolysis, and oxidative metabolism. This was all about the energy sources required for muscle contraction.
Introducing sqadia.com on your mobile, making medical education more accessible than ever. Your one-stop shop for medical learning now fits in your pocket. Visualize, understand, and master as one seamless process. Access exclusive courses on anatomy, physiology, and more, right from your phone. With intuitive navigation and engaging video content, your learning becomes enjoyable and effective. Get started with our free trial. An abundance of educational resources is now just a tap away. Download now the sqadia.com app from Google Play or the Apple App Store. sqadia.com. Medical education at your fingertips.